Thank you. Thank you for the invitation. It's a pleasure to be here and present the work we do in the lab. So our main question is how the cellular microenvironments are shaping cellular identity and response. And to, to resolve this question, what we do is to use single-cell genomics and imaging tools to profile the tissues in vivo, and then we do computational predictions that we then validate using in vitro models. So central to our main question is the development of computational tools, and particularly this CellPhoneDB, which is a computational tool to look for cell-cell interactions from single-cell data that I developed during my postdoc in the Teichmann team. And what is unique about this method that is based on a repository of ligand interaction and ligand and receptors and their interactions is that it's a really well curated data set, and also that it comprises these complexes. So, well, we know that the majority of receptors and ligands also are part of have multiple subunits, and you need multiple of them to be co-expressed in order for this receptor to be functional. And this is a particularly important one for all the processes, and in our case for development and immune system that we are interested. And that was what is stored in and considered in our data. And basically we then do we then look for ligands and receptors on the single cell data. We were doing random shuffling to look for a specific interactions. So moving forward in our team, we are continue developing this CellPhoneDB, and we have recently integrated these predictions with a spatial transcriptomics data, where we just consider that cells that are in the same microenvironment and that give us the opportunity to improve our interactions. And unpublished data also from our team is a new version of CellPhoneDB, where not only we have improved or store more interactions or more curated interactions, including those that are small molecules, and in this case, because they are non-protein products, we look for the synthetic pathways to build them. But also we have curated several receptors, ligand, and transcription factors that are part of the same pathway. And these give us the opportunity to give us a subset of interactions that are more likely to be happening, because we use this transcription factor that could be predicted by transcriptome, but also by a tag as a sensor of this pathway. So for today's presentation, I'm going to tell you how we have used CellPhoneDB and other tools to study the human endometrium. So, well, we are interested in development, but in this case, we were not looking at the developing embryo, but instead we were looking at the uterus, that is the environment where this embryo develops. So the endometrium is an epithelial tissue that, in comparison to other epithelial tissues in the body, is poorly studied. So this endometrium, this epithelium from the endometrium has this luminal epithelium, will establish communication with the developing embryo, and also have these invaginations or glands that are full of secretions and that will provide the nutrition to the embryo during the first stages of development. So what is quite interesting and got us fascinated about this tissue is the regenerative capacity. So every month, the endometrium goes to cycles of shedding, regeneration, proliferation, regeneration, and differentiation. And this is mediated by the two ovarian hormones, estrogen and progesterone. So during the first proliferative phase, that is dominated by estrogen, then we have the ovulation, and then progesterone levels start increasing, and these tissues start differentiating. And in simian primates, This happens in the absence of embryo implantation. And this really highlights how different this process is, for example, with mouse, and the importance to study the biology of this tissue in humans. So because we don't know that much about this tissue in humans, we ask very basic questions. So what are the cell types that make up the endometrium and how they change across the menstrual cycle? What are the key signaling pathways that will control this endometrial proliferation and differentiation? And finally, we ask ourselves if we can use the current in vitro models and organoids to validate those. And while we came here from a very basic biological questions, then when we started the work, we also realized how our work will have an impact on women's health, and in particular, for example, for endometriosis. I just want to mention endometriosis is a painful disease that is suffering 1 out of 10 women in reproductive age. And it's quite surprising that there are not good or non-invasive diagnostic tools and either treatment for these women. And in part is because we have a very poor knowledge about the biology of the tissue. So the first thing we did is to profile samples. And in particular for this cohort, what we were sampling is not only endometrial biopsies that takes the superficial layer, but also full uterine samples that came from the organ donor program. So people who die from non-gynecological disorders. And then we profile our 10 individuals with other individuals that were published recently in a Nature Medicine paper. And then we integrated all the data sets and we find the main populations. So we managed to define a lot of main populations and subsets. And just as a side story for this, I'm quite excited because in the immune part, we defined the 3 NK cells that I previously defined during my postdoc in the uterus during pregnancy and interactions with the placenta. But this is not that was anecdotic, and I don't want to distract you on that, because for this work, we are focusing on the epithelial cells. So this tissue, it changes a lot during the proliferative and secretory phase. And as you can see, the composition is changing. So in the proliferative, we have these SOX9 populations, which we define 2 populations depending on the expression of LGR5. And then we have in the more secretory phase or differentiated phase, the luminal, the glandular secretory, and the ciliated. So because we define these SOX9 populations that depend on the expression of LGR5, we then wanted to see where they are located. And to do that, we use spatial transcriptomics, and then we use cell to location to integrate it with single cell and deconvolve the wells. And what we can nicely see is that the LGR5 populations are on the lumen, while the LGR5 negative populations are in the basal layer, and we suspect that these luminal ones are giving rise to the populations in the lumen, while these ones will be more to the ones in the glands. And we have also validated these using high resolution microscopy. So with this map, this is all in healthy samples, and then we wanted to see if we can use that information to understand better endometrial pathologies. And basically we analyze endometrial cancer and also endometriosis, and in both cases we see an enrichment of the SOX9 positive population. And just this is also quite a nice finding in the endometrial cancer, because we have 2 types, the serous and the endometrioid. The serous is more aggressive, and we see the signature. This is just the convoluting bulk data with single cell, and in this case, we see the signature of LGR5 positive progenitor, while in the endometrioid endometrial cancer, we see that half of them are LGR5 positive, while the other half are LGR5 negative. And actually, the ones that are LGR5 positive is correlating with more advances of this cancer. So with that, I'm just highlighting how these pathologies are enriched on the progenitors. It really highlights how important it is for them to have a these epithelial cells to properly differentiate. And that's where we started to look for the differentiation pathways by measuring the transcription factors and also their activities just by looking at the genes that are known to be regulated by these transcription factors. And then what we see is that in the epithelial cells that will differentiate towards the ciliated lineage, the Wnt pathways become active, while the ones that differentiate to the secretory lineage, it's an inhibition of Wnt and then an activation of Notch. We wanted to see if this pattern of the pathways is also seen in space, and we do that again by using spatial transcriptomics. Here we didn't integrate it with single cell, we just did clustering, unbiased clustering on the epithelial cells, and you can nicely see how just the unbiased clustering really clusters with the lumen and the glands and the base are just highlighting how important is the environment for the identity of those cells. And again, we see the same pattern that more in the lumen, where it's enriched in ciliated cells, we see Wnt inhibitory signals, while in the glands, that is more enriched in secretory cells, is more transcription factors that are known to be inhibited by Wnt and transcription factors that are known to be active by Notch. And we just also validated signals for Wnt and Notch just using high resolution microscopy and quantifying, and just here showing the high presence of Notch in the glands in comparison to the lumen, and also it is quantified here. So we of course wanted to study the signals from Wnt and Notch, and to do that we use CellPhoneDB, the one that is integrated with the space. And we divide it in proliferative and secretory phase. And well, in the proliferative phase, we see that Wnt is active kind of everywhere, and that makes sense because it's essential for proliferation. But then in the secretory, in the cells that are the secretory lineage and the ones in the glands, they downregulate Wnt receptors, and also these stromal cells or endometrial stromal cells that are fibroblasts that intermingle with the glands express DKK1, which is a potent Wnt inhibitor. So with that, really from this single cell profiling, what we see is this disbalance between Wnt and Notch happening in different spaces of the tissue, in the glands, on the lumen, where it's enriched on different epithelial space, the secretory or the ciliated. So we wanted to really validate our findings and understand how this disbalance between Wnt and Notch can affect epithelial cell differentiation. And to do that, we use endometrial organoids, together with Margarita Turco from Cambridge University. And what is quite nice from these organoids is that you derive them from primary tissue, and then once you have established them, you can stimulate them with hormones, with estrogen and progesterone, and they behave in theory as the in vivo epithelial cells in the endometrium. So we profiled them because the first thing was to benchmark the model. We profiled by single cell. By markers, we can annotate the different ciliated and secretory lineage. But of course, we wanted to kind of benchmark it in a more quantitative way. And to do that, what we did is to build a logistic regression model using our in vivo epithelial cells, and we project it into the organoids. And what we see is that, well, just want to mention here, we just consider the cells that are induced by hormones, because in vivo you always have hormones. And we can nicely see that upon estrogen, we have the SOX9 populations. And then when we put the progesterone, we have the ciliated lineage popping up and these preciliated populations that we identify. And then we have the secretory, and in the secretory lineage, we have some SOX9 that are becoming differentiated into the secretory one. So we don't care, to be honest, that much if the organoid is not fully differentiated or not, which as you can see in the secretory is not fully, it will be around 40% of the secretory cells. But what we care about is if those epithelial cells are responding in the same way that they in vivo, because we want to understand the pathways for differentiation. And to do that, again, on these subsets, we calculated the transcription factors and their activities, and we could nicely validate how the same transcription factors that we show in vivo that are responding in the ciliated lineage pop up here, and exactly the same happens with the transcription factors for the secretory lineage. So therefore, we have a nice system where we can validate the effect of notch and wind, and that's what we did. So here, in addition to put the hormones, we combined them with 2 inhibitors for notch and 2 inhibitors for wind, and we performed single-cell transcriptomics data on different conditions, and in addition, we do some immunostaining. And I'm just going to show you how nice the phenotypes were. So basically, when we put the notch inhibitors here, we can nicely see how we don't have secretions at all, and then we have a lot of ciliated cells, and this was also recapitulated by a single cell. And actually, the completely opposite pattern happens with the Wnt inhibitors, when we have very few ciliated cells and we have loads of secretions. So then the nice thing about profiling with single-cell transcriptomics is that in addition to looking at the efficiency, which was really validated for the notch and wind, we can also look for the accuracy. And that was relevant in the condition where we put Wnt inhibitor, because for the notch inhibitor, we don't see a new subset coming up, but we do see a new subset in the Wnt inhibitor. And actually, we wanted to see, is this subset producing more secretion? And that's what we did here. And actually, you can nicely see how this is the case. So when you have hormones and Wnt inhibitor, then you have more secretions. And we validated this also at the level of protein. And this is in vivo is happening because you have the stromal cells that will induce this Wnt inhibitor environment on top of the progesterone. And then we can also dig more into the mechanisms and the relationship between hormones and pathways. And for example, one of the things that we realized that Wnt inhibitor, if you put it on their own, it doesn't induce the secretions. And this is just because the hormones are required to, for example, activate notch that is not activated if you only put a Wnt inhibitor. And with all of that, we have built this model using single-cell and spatial methods and this nice resource that is publicly available, and we have a nice interface to visualize it, where we can see how cell stage change across the menstrual cycle, and then we also define these 2 environments that have a disbalance between wind and notch. And well, that's all for us. I just want to acknowledge the 2 members in my team, Luz Garcia Alonso and Louis Francois Hansfield, who led this project. This project was also a collaboration with the Sarah Teichmann team. That's because I started this project while I was a postdoc in her team, and also with Omer Bayraktar for all the help in the imaging, and Margarita Turco for the help on the organoids, and of course the donors. And thank you so much.
Thank you, Roder, for a super interesting talk. We have a question from Arjun Raj. Very nice work. It's great to see the environment and effects of space. Do you think the reason for the differences has its origin in differences in the protein composition of the ECM, or by physical differences, like different tensions on the cells in these different locations, or something else entirely?
You mean, sorry, between the organoids and the in vivo data, or?
I'm not sure, between the organoids or between the different stages or between the different zones, like the ciliated zone or the secretory zone. So he's wondering about the role of the ECM tension versus maybe morphogens, or.
Yeah, that's correct.
I hope I get it right, Arjun.
Yeah, I mean, we of course simplify it to the Wnt and notch, but there is quite differences because, for example, in the lumen, you don't have that much of a connection with these stromal cells where you have the glands embedded with, and that really it's or not only the pathways, but of course also the extracellular matrix proteins. But we have just reduced it to this winter node signaling. But yeah, there are also a lot of different interactions that we haven't explored so far.
Thanks.
Okay, so the next question is from Yotam Harnik, who says, thanks for the interesting talk. How do you think the balance between the two lineages is regulated?
Well, yeah, I do think that is really because the spatial position that you have into the space, because, well, and that's why it is a balance. It's not that in the lumen you only have ciliated cells and in the glands you only have secretory, but because in the glands you have more access to a Wnt inhibitor environment, then you can really differentiate towards the secretory lineage, while in the lumen, where you don't have this access to that much of a Wnt inhibitor, then you differentiate towards the other lineage. So, but again, it is a balance because it is really an efficiency thing. It's not that it's black and white, if that makes sense. So it's really the spatial position that I do think that will change, and the interactions that this spatial position is associated with.
Thanks. We have another question from Maria Bermudez. Do you know if this notch-Wnt signaling relationship can be changed in pathological conditions such as cancer?
Yeah, I mean, that's a really interesting question and something that we have been also exploring. And for example, we have been looking for GWAS, and we do see on the GWAS a lot of hits really are related to the Wnt and notch, and also the crosstalk between the fibroblast and the epithelial cells. So certainly, I'm pretty convinced that there will be something related to that. But of course, we will need to explore further using pathological samples. So this is just crossing the GWAS hits with our powerful kind of defined single cell data set from healthy samples.
Okay, one more from Joshua Thiessen. Have you performed RNA velocity analysis on your data? One of the first UMAPs you showed had a somewhat circular appearance, similar to the cell cycle UMAP shown in one of the talks yesterday. Yeah, I mean, the problem with velocity for the organoids, yes, and we can reconstruct it easily. The problem with the adult samples is that they come from a lot of individuals. So it is really difficult to build trajectories on them, if that makes sense. And using velocity and all these tools. So yes, for the organoids, that's simple. But for the in vivo data, it is, and we have also done clonal organoids, so derived from one single cell. So we are sure that this progenitor can differentiate into both of them. But for the in vivo data, yeah, it is, because you never have the progenitors and the differentiated cells on the same individual. It really changes, and that's why it is really difficult. I wouldn't say impossible, but at least in our hands, it was almost impossible.
Okay, there's a very interesting question here from Timothy Powell. Is there commonality in gene expression differences between the ciliated and secretory epithelial cells in the endometrium and these epithelial cells in lung or intestine? Can you see a common signature for ciliated cells in different locations or tissues?
Yeah, that's correct. We haven't done a comprehensive analysis, but actually they are quite similar with the ones in the lung. We have done some analysis, and you do see a lot of similarities between the epithelial cells in the endometrium and the lung, and also this Wnt and notch regulation is similar. It's not exactly the same because some stem cell populations just in the lung, you actually don't see them in the endometrium. So not exactly the same, but a lot of common pathways on the ones in the lung.
Okay, I think we have about one more minute, so let's squeeze in one more question from Jingqi Wang. In the cell phone DB, could you quickly explain how you take into the concentrations combinations of ligands and receptors to predict the active pathway? So how do you take into account the concentrations combinations of ligands and receptors to predict?
Okay, I'm not sure I am following that question. So do you mean So we just consider, in self-NDB, we just consider the expression of receptors and ligands. And what we did is, well, the point is a lot of ligands and secreted ligands we have done, they are very expressed by very few cells. So that's where, I mean, the user can decide, but we usually say that around 10% of cells need to express that. And then the random shuffling, what allows us is to look for a specific, but actually you can also run the same, and we are running the same with differential expression genes to prioritize, but this is more to prioritize the interactions that are specific. I don't know if that's what the question was.
I think that makes sense. Okay, well, thank you, Rosa.